Okay, I'd like to begin by thanking David and the organizers for the opportunity to be part of such an exciting symposium. In the time I have this afternoon, I'd like to talk about an ongoing project in my lab that we've been focused on for some time now, looking at p53 as a signaling pathway in human cancer, and in particular, in a very rare childhood cancer called adrenal cortical carcinoma. So just by means of some introduction to the tumor that we're studying, the adrenal glands sit on top of the kidneys, and the adrenal is divided into the inner part, which is the medulla, the cortex, and then the capsule. And it's the cortex that gives rise to the tumors that we're studying. And when the cortex becomes tumorigenic in children, it's often hyperactive where it's overproducing these adrenal hormones, which regulate blood pressure, glucose metabolism, immune competency, and sex development. And so these children who develop adrenal cancer often come to the clinic usually between 3 and 4 years of age, and it's more frequent in girls. And they present with virilization where they'll have facial hair, they'll have developed pubic hair, body odor, acne. They've gone through puberty in every way. They'll have high blood pressure, muscle fatigue. They'll have sometimes quite large tumors. And it's often within the context of Li-Fraumeni syndrome. And so our project began in a city called Curitiba in southern Brazil, in the state of Paraná. It's south of Sao Paulo. We originally studied 36 children who developed adrenal cancer. And 35 of those 36 children all inherited the same p53 mutation, which is 337 histidine. First thing we did was to try to understand the consequence of this mutation on p53 function. So we engineered the mutant, we overexpressed it in a variety of different cell types, addressing it in a variety of different p53 assays. And in every case, 337 behaved just like wild-type p53, whether it's promoter-reporter assays, apoptotic assays, or the ability to just block a cancer cell from growing altogether. And it behaves just like wild-type p53 and not like a DNA-binding mutant that would be associated with Li-Fraumeni syndrome. So I want to emphasize the point here that 337 is different than your typical DNA-binding mutation in p53. But we knew there was a problem with this mutation when we looked at the tumors. The tumors always undergo LOH. They delete the wild-type allele, and they duplicate the mutant allele. And that mutant allele is active, it's expressed, and the missense protein is at high levels accumulating in the nucleus. If it was doing its job, it should be blocking the growth of those tumor cells or inducing the cell death of those tumor cells. Clearly, it's not. So just to summarize, a lot of different studies, work done by our group and by other groups here in the States and in Brazil. What we know is that almost every child from Brazil who develops an adrenal cortical carcinoma have inherited a germline p53, the same p53 mutation corresponding to arginine 337 histidine. Amelia Pinto, working with Claudia Latronica, showed that it was a founder mutation. And then work by Pierre Hanot and Maria Isabel showed that that founder was likely a European Caucasian carrier that brought that mutation from Europe to Brazil probably several hundred years ago. We developed a PCR test in our lab that allowed us to easily screen just for this mutation. That allowed our collaborator, Bono Figueiredo in Curitiba, to screen 175,000 newborns. And what he found that 1 in every 375 have inherited this p53 mutation. This includes the regions of Sao Paulo and Rio de Janeiro, a population of anywhere from 80 to 100 million people. And 1 in every 375 have this germline p53 mutation. It's clearly a significant health issue facing our friends in Brazil. Our functional assays failed to show a defect. but we know from structural analysis that the mutation has a consequence on its ability to form stable dimers and tetramers, and it's a pH-dependent effect. We know the tumors always undergo LOH, selecting for the mutant allele, overexpressing the missense protein at high levels in the nucleus. And what's very important to keep in mind is that if you're born with this mutation, your risk of developing adrenal cancer is anywhere from 1 in 10 to 1 in 30. The normal incidence is 1 in 3 million. So you can see how it's raised your risk for this cancer in a very significant way. Now, the good news is 9 out of 10 or 29 out of 30 individuals who are carrying this mutation don't develop this cancer. So in that initial study where we looked at 36 children with adrenal cancer, they came from 30 different families. 23 of those families had many carriers of this mutation, and they were fine, at least not cancer prone. But 7 of those 30 families, you could identify young women with breast cancer, someone with a brain cancer at a young age, much more like Li-Fraumeni or Li-Fraumeni-like syndrome. And again, follow-up work by Pierre and Maria Isabel showed that you can actually screen for patients with Li-Fraumeni syndrome and find an association of this germline mutation in that context. So the question is, how can you have a founder mutation, so it's the same exact mutation in all of these carriers, and have some families that are at low risk for cancer, and some families at high risk for cancer? So we wanted to try to understand how that could be, and so we developed a genetically tractable model where we knocked the mutation into the mouse. And so we just used the conventional approach for introducing the single point mutation at codon 334 in the mouse, which is equivalent to 337 in humans. First set of experiments that we did with these mice is we looked at the ability of the endogenous 334 to work like P53, a wild-type P53. And that's where we took thymocytes and treated them with ionizing radiation, in this case, 5 grays. So if we have wild-type thymocytes untreated, these cells are viable, healthy, they can maintain that viability for an extended period of time. But if we treat those cells with ionizing radiation, they undergo massive cell death. If you do the same experiment with thymocytes from a mouse that has no P53, the untreated cells are viable, and even after treating them with 5 grays, they maintain their viability. The 337 mutant thymocytes are in between. They're clearly more resistant than wild type. They're clearly more resistant to DNA damage to induce apoptosis than wild-type thymocytes, but not as good as P53 knockouts, suggesting that it has partial activity. And we can see that when we do Western blot analysis. If we treat wild-type thymocytes with DNA damage, you see an induction of P53 and an induction of P53 target genes such as P21. If we look at the homozygous 334 mutant thymocytes, they already have abnormally high levels of the missense protein in untreated thymocytes. DNA damage induces it slightly, and even though you have this abundant amount of 334 protein, much higher than wild type, its ability to induce P21 is drastically reduced. So it says that it's partially functional, clearly not as competent as wild-type P53. And then what was also surprising to us is that the 334 mutant in the untreated thymocytes was even higher than the DNA-binding mutant 172, which is the Li-Fraumeni mouse made by Gigi Lozano. And it was shown by by Western blot and also by immunohistochemistry, where we can see that the missense 334 protein is in the nucleus of these thymocytes. So it's abnormally expressed, it's functionally compromised, but not inactive. We increased the dose to 10 grays and essentially got the same result. The untreated thymocytes, this is in vivo, The 334 protein is at high levels in untreated thymocytes, no real effect after DNA damage, and it's compromised in inducing P21. This is just some immunohistochemistry showing that the missense protein is detectable in the untreated GI tract, in the crypt of the GI tract, and it's higher than what we see in the 172 homozygous intestine. It's true not just in mice, but also in people. So here we have an individual from Brazil who developed adrenal cortical carcinoma. And when we do immunohistochemistry, we see high levels of the 337 missense protein in the nucleus of the adrenal tumor. And then if we look at the adjacent normal adrenal, we see the same thing, where there's high levels of this missense protein in the nucleus, indicating that it's just naturally, abnormally expressed at high levels. We extended these experiments slightly further, where rather than just looking at specific target genes, we're looking at gene expression as a whole. So this is just some Affymatrix gene expression profiling. We show that if we treat wild-type thymocytes with ionizing radiation, a series of genes are upregulated, another series of genes are downregulated. And if we look at thymocytes from the 334 mutant mice, you can see that these genes are compromised in their ability to be induced or to be repressed. And if we just focus in on P53 target genes, we see that these target genes are upregulated by wild-type P53 and severely dampened by the 337 mutant. And we've quantified these results by real-time PCR just to determine the quantitation. So what's this mean for the mice? You have abnormal expression of 337. It's functionally attenuated in turning on target genes. It's impaired in its ability to induce cell death. So if we just let these mice age, they do develop cancer. But with incomplete penetrance, only about 20% of the mice get cancer. And they get tumors at late age, so a delayed onset, usually after about a year and a half. And I want to contrast that to what we know with the typical DNA binding mutation that you find in classical Li-Fraumeni. The 175 histidine mice all develop cancer, essentially, usually between 3 and 6 months of age. The 337 mice are somewhere in between the DNA binding mutant and wild-type mice. So just to summarize the mouse model, we've generated a mouse model that represents the most common inherited P53 mutation, the 337 mutation. It's abnormally expressed, functionally compromised. We call that hypomorphic. And despite it being abnormally expressed and functionally compromised, the mice are largely resistant to tumors. They are susceptible, but not like a DNA binding mutant. And we think that's consistent with the variable penetrance that we see in the carriers from Brazil. So that leads us to think that 337 raises your risk of cancer, but mildly so, and that there must be something else that cooperates with that mutation to lead to that Li-Fraumeni type of phenotype that you see in those high-risk Brazilian families that have this mutation. So we tried to address that issue by doing a comprehensive genomic characterization of these pediatric adrenal tumors. We've done whole genome sequencing, whole exome sequencing, RNA sequence analysis. And we focused on 19 adrenal tumors and 19 matched blood samples to look at what the differences are. Included in that group of pediatric adrenal tumors are 11 tumors from Brazil, all associated with the 337 mutation. And what we found was that, in fact, all 11 Brazilian adrenal tumors had the same founder P53 mutation, the 337 mutation, as you would expect. What was surprising to us, though, was that 4 of the tumors out of the 11, in addition to having the 337 mutation, carried a second mutation in an adjacent gene called XAF1. So P53 is on chromosome 17 here, and approximately 2 centimorgans away is this gene called XAF1. And we find a nonsense mutation in XAF1 in 4 of the 11 Brazilian tumors. And this XAF1 mutation is in and of itself a founder mutation. So here you have a situation where you have 2 founder mutations that have come together that perhaps can increase risk of cancer. So a little more background about XAF1. It was initially identified in a yeast 2 hybrid screen where they were using the survival factor XIAP1 as bait. So XAF1 binds XIAP1. It's a 301 amino acid zinc finger protein that's ubiquitously expressed, including in the adrenal gland. And what's important to note is that XAF1 is selected against in a variety of different human tumors, and it's selected against by epigenetic control. And then some very important work by Li and coworkers showed that XAF1 is actually a target gene of P53. So P53 induces XAF1, and XAF1 functions within a positive feedback loop with P53. And so Li and coworkers says that XAF1 will bind P53 and will bind MDM2, and in so doing results in P53 stabilization, activation, and enhanced tumor suppression function. They also showed in a very elegant way that if you overexpress XAF1 or knock out XAF1 through loss of function studies, that its activity is consistent with being a tumor suppressor. Again, its expression is being selected against in a variety of human cancers. So here we have a nonsense mutation existing in the germline of these patients. So it's a proapoptotic tumor suppressor. We followed up Li's work to see whether XAF1 and the nonsense mutation had any effect on P53 function. And so here we have just a promoter-reporter assay which has low basal expression. You can add XAF1 wild type or XAF1 nonsense mutation. It has no effect on promoter-reporter activity. If we add the 337 mutant P53, 337P53, it induces this promoter, inducing reporter expression. And then if we co-express 337 plus XAF1, it enhances activity significantly. And if we use the nonsense mutation, it does not. And in fact, if we add wild-type XAF1 plus the nonsense mutation acts as a dominant negative to prevent it from enhancing 337 transcription. So this leads us to the hypothesis that loss of XAF1 further compromises 337 activity, which leads to the increased risk of cancer. So we've been able to determine the allele frequency of these mutations, both the 337 mutant and the XAF1 mutation. 337 exists primarily in Brazil. We know of families in Europe, a few families that will have this mutation. But 337 is largely restricted to Brazil. Again, 1 in every 375 individuals. The XAF1 nonsense mutation from databases shows that it's a polymorphism that exists in Europe. And we screened 3,000 newborns for this mutation in Brazil that were negative for 337. And we found essentially the same allele frequency in Brazil. And this allele frequency calculates out to be about 1 in every 130 Brazilians. So if we just now start screening for these 2 mutations in tumors arising in Brazil, tumors that are associated with the 337 mutation, what we find is that 92% of the sarcomas will have both mutations, which is consistent with the hypothesis that XAF1 is influencing p53 activity, leading to the higher risk of cancer. And here we have just one pedigree of a Brazilian family where the child presented with the adrenal tumor. We identified the 337 mutation in this patient, and we found that this mutation exists in multiple family members, some 80 years of age or greater, and it's only the 337 mutation, consistent with it being at a lower risk of cancer. And then we have another Brazilian family where we've identified patients that have developed sarcoma. Each of the individuals who develop sarcoma have, in addition to the 337 mutation, the XAF1 nonsense mutation. And then there's 2 carriers at younger age that haven't developed cancer, but they are adolescent or younger. So to summarize this part of the talk, we've identified an XAF1 nonsense mutation. It's a founder mutation. It's linked to the 337 mutation in Brazil. It's a proapoptotic tumor suppressor, works within a feedback loop with p53. And we propose that the XAF1 mutation in cooperation with the 337 mutation is what leads to the increased risk of cancer. And we're now modeling the XAF1 mutation in our mice. And I'll stop there and just want to thank all our collaborators for their help.
A couple of questions, and then Gigi, start. Just, yeah.
So there are some Li-Fraumeni families and Li-Fraumeni-like families that don't have p53 mutations. Do they have XAF1 mutations? Have you looked?
We haven't looked, but I would say that's a possibility. My feeling, though, is XAF1 alone is not going to be driving cancer at a really high rate. It is a polymorphism that's pretty common, worldwide.
Do you think, that was a fantastic talk, amazing data. Do you think that there's a special relationship between XAF1 and that particular p53 mutation, or do you think it could be modulating the effects of other p53 mutations?
Yeah, really good question. We don't know the answer to that yet. This is really early data that we've generated. But the way I think about it is that if you have a DNA binding mutant like 175 histidine, it's broken. And I don't know whether you had an XAF1 nonsense mutation, whether it would make it any different than what it is. I think it probably plays a more impactful role in those hypomorphic mutant p53s. Carol?
So the mutation itself is in the oligomerization domain. What sort of defect in oligomerization is known about this R337 mutation? And could that explain why you have to lose the wild type, because it can't do anything to the wild type? Is that right?
So that's how
co-tetramerize with the wild type, right?
Correct. So the question was, what's the consequence of the mutation on dimerization, tetramerization, and whether it could actually play a role as a dominant negative kind of mutation? So work that we did with Richard Kawacki a while ago showed that changing the arginine to the histidine, it's right at a salt bridge. So arginine is at the beginning of this alpha helix, and at the end of the alpha helix is an aspartic acid, and the dimer comes together in opposite directions. So it places the arginine over the aspartic acid. That forms the salt bridge. So it's like 2 clamps holding p53 together. When you change arginine to histidine, histidine can be charged, the imidazole ring can be charged, but under acidic conditions. Even when it's charged and forms that salt bridge, it's not as stable. It's like 20 degrees more unstable than wild-type p53. And then if you get to neutral pH and slightly basic pH, then it's not even charged, and so the whole thing can fall apart. So it doesn't act as a dominant negative, and that may be part of the reason why you need to delete the wild-type allele. And we have data to say that its ability to form dimers and tetramers is compromised in vivo, and we think that's the reason why it's abnormally expressed, in normal tissue that hasn't been stressed. It's not forming stable tetramers that are usually what's recognized by MDM2.
So isn't that a kind of amazing that it has such normal activity when it's expressed ectopically? It can't form any kind of a tetramer, right? Or even a dimer, if I got it right.
We worked long and hard trying to see any kind of consequence. If you make a cDNA, overexpress it, driven by a CMV promoter in any cell type you look at, in any p53 assay you look at, it was always like wild type. That's why we wanted to do the mouse model where we could study it under a more physiologic condition.
I think we're going to have to.
Okay.
Thanks very much, Gerry. Thank you. Amazing.